Good day everyone, I'm Samantha Ben-Ami Barrientos, a second-year student of Bachelor of Secondary Education, major in Science, and for today, we will talk about the nucleic acids, how structure conveys information. For the introduction, nucleic acids are biomolecules that act as a blueprint of life, specifically in DNA or deoxyribonucleic acid and RNA or ribonucleic acid. The structure of DNA with its double helix made up of complementary base pairs enables it to store genetic information in a stable and organized way. Each sequence of bases, the adenine, thymine, cytosine, and guanine, represents coded instructions that determine an organism's traits. Meanwhile, the RNA plays a key role in translating this information into proteins which carry out the cell's function. The differences in structure between DNA and RNA, such as the sugar component, nitrogenous bases, and the number of strands. That's what makes them different, and each molecule is suitable for its specific role in the flow of genetic information. Ibig sabihin lang nito, the structure of nucleic acids is not just about chemistry. It is what makes heredity, evolution, and all the biological processes possible. So, this will be the contents included here in this video presentation. First is the DNA denaturation. It is the process where the two strands of the DNA double helix separate. This happens when the hydrogen bonds that hold the base pairs together and the stacking interactions among the bases are disrupted. Ibig sabihin, kumbaga nagkagulo. Usually galing sa heat. So as the DNA is heated, the double helix unwinds and the strands become single-stranded. Now, how do we monitor DNA denaturation? It can be observed through ultraviolet light absorption at a wavelength of 260 nanometers. As the DNA strands separate, the absorption of light increases. This increase in absorbance is called hyperchromic effect, which occurs because the bases become unstacked during denaturation. This process is reversible. When we say reversible, ibig sabihin, kapag na-pull na yung DNA, the separated strands can rejoin with their complementary bases to reform the double helix. The process is known as renaturation or annealing. Let's proceed to the principal kinds of RNA and their structures. Inside the cell, there are many types of RNA, but the three main kinds are mRNA, rRNA, and tRNA. Each one plays an important role in the process of protein synthesis. All RNA molecules are produced from DNA through a process called transcription, where the sequence of bases from DNA is copied into RNA. So, first, we have the messenger RNA or mRNA. This type of RNA carries the genetic code from the DNA in the nucleus to the ribosomes. It acts like a blueprint that determines the exact sequence of amino acids that will form a protein. Next is, we have the ribosomal RNA or rRNA. Together with proteins, it forms the ribosomes, which are the actual sites where the proteins are built. Think of ribosomes as the factory where proteins assembly takes place. Lastly, we have the transfer RNA or the tRNA. This RNA delivers the correct amino acids to the ribosomes during protein synthesis. Each transfer RNA is linked to a specific amino acid and matches it with the corresponding codon or a group of 3 bases found on the messenger RNA. So, the process where the base sequence of mRNA directs to the sequence of amino acids in a protein is called translation. So, to summarize all of that, messenger RNA carries the message. Ribosomal RNA builds the site, and the transfer RNA brings the materials. These 3 types of RNA ensures that proteins are made accurately based on the genetic instructions stored in our DNA. Let's proceed to the new age RNA types, the more recently discovered kinds of RNA that go beyond the 3 primary types. First, we have the small nuclear RNA. This type of RNA is found only on the nucleus of eukaryotic cells. Its main role is to help process the newly formed RNA to its mature form before it leaves the nucleus for translation. Next are the microRNA and small interfering RNA. These two plays a very important role in a process called RNA interference or RNAi, which helps control or silence specific genes. This process was first discovered in plants but is also present in animals, including humans. The microRNAs also help to protect bacteria from viral infections and assist in repairing nerve and muscle damage. Another exciting discovery is the long non-coding RNA. These are RNA molecules that are longer than 200 bases but do not code for proteins. For a long time, scientists thought that they were mRNA or the messenger RNA, but now we know that they serve different purposes. These long non-coding RNA have been linked to important biological processes such as the silencing of 1X chromosome in females and even to certain diseases. So, in summary of these 3, the discovery of this new age has shown us that RNA is much more than just a messenger. It plays a diverse and essential roles in gene regulation, development, and health. Let's proceed to the roles of different kinds of RNA. First is the transfer RNA or the tRNA. Transfer RNA is a small type of RNA found in all living cells. It has a clover leaf-like structure formed by base pairing within the strand. Each transfer RNA carries a specific amino acid and has an anticodon that matches with the codon on messenger RNA, ensuring the correct amino acid is added during protein synthesis. Second is ribosomal RNA or rRNA. Ribosomal RNA is one of the most abundant and largest types of RNA in the cell. It forms the main structural and functional parts of ribosomes together with ribosomal proteins. rRNA helps maintain the ribosome shape and ensures the proper alignment of messenger RNA and transfer RNA during protein production. Third is messenger RNA or mRNA. Messenger RNA is a variable-sized, single-stranded RNA that carries genetic information copied from DNA. It serves as a temporary template that determines the order of amino acids in proteins. Messenger RNA is a short-lived as it is only made when needed and quickly broken down after use. Fourth is small nuclear RNA or snRNA. Small nuclear RNA is a short RNA molecule found only in the nucleus of eukaryotic cells. It combines with proteins to form snRNPs, which are involved in RNA processing. It plays an important role in editing and preparing for pre-mRNA before it becomes mature and ready for translation. Fifth is small interfering RNA. Small interfering RNA is a short double-stranded RNA molecule. It naturally occurs in cells and helps regulate genes by binding to complementary mRNA molecules. This binding leads to the degradation of the target messenger RNA, preventing them to produce a protein. Sixth is the microRNA. It is a small non-coding RNA that helps regulate gene expression. It binds to specific mRNA targets to block their translation or trigger their breakdown. These are especially important in controlling cell growth, differentiation, and development. Lastly, long non-coding RNA. It consists of long RNA molecules that do not code for proteins. They are found in the nucleus of cytoplasm and can regulate gene activity at different levels. Although the function of these are still being studied, but the long non-coding RNAs are believed to play roles in the development of certain diseases.
For our last topic, let's proceed to the RNA and medical applications. Scientists have discovered different types of RNA that can be used to study, treat, and even prevent diseases. First is microRNA. MicroRNA was discovered in 1993. These are short RNA segments that can bind to messenger RNA to stop it from being translated into proteins. In simpler terms, microRNA acts like a gene silencer, preventing certain genes from producing proteins that may cause disease. One example of its application is the drug miraviricin, which is used to treat hepatitis C. This drug works by blocking the virus from replicating inside the body. Itong microRNA is pinag-aaralan din sa cancer research kasi nakokontrol nito ang cell growth and apoptosis na nakakalesan sa process ng tumor sa katawan. Next is we have small interfering RNA. It is very similar to the first one and how it silences genes, but this RNA usually targets only one specific gene. Researchers have tested small interfering RNA-based drugs for diseases like the Ebola virus. In those studies, this helps stop the virus from reproducing, improving the survival rate of infected monkeys during trials. Itong medical technology na ito is ginagamit na rin para sa human medicine. One example is the drug patisiran, which treats a rare nerve disease called hereditary transthyretin amyloidosis. It's one of the first base therapies approved for clinical use. Third is clustered regularly interspaced short palindromic repeats. This is a DNA sequence found in bacteria that works together with proteins called Cas, acting as a defense system against viruses. But in modern medicine, scientists have learned to use this as a gene editing tool. Researchers can cut and replace faulty genes in living organisms. This technology is currently being studied to treat genetic diseases like cystic fibrosis, sickle anemia. Itong medical technology na ito ay very efficient because it's fast, accurate, and affordable compared to older gene editing methods. Lastly, we have long non-coding RNA. This is a newly discovered type of RNA that doesn't code for proteins but still plays major roles in regulating gene expression. These are found throughout our cells and are linked to many cancer types. Ginagamit itong early cancer detection since their levels often change when cancer cells form. To summarize it all up, RNA isn't just a messenger for DNA. It is a powerful tool that scientists use to understand, prevent, and treat diseases. RNA technology continues to shape the future of medicine. Thank you for listening.